Dear colleague, dear students, this is the second part of a course on biodiversity informatics data. This part is subdivided into two subparts. The first one concerns the primary biodiversity data, and the second one concerns the workflow for quality check of primary biodiversity data. The primary biodiversity data are characterized by four attributes. The first one is the taxon, which is, let's say, the name of the organism that is observed. Here, the name is the scientific name provided by a taxonomist and a specialist in the field of botanic, for instance, for plants or animal taxonomist with respect to, let's say, the animals. Anyway, it should be an updated name, scientific name. And when the taxon is identified to species level, then it is quite interesting. Otherwise, the genus or family level identification are better than nothing. The second attribute is the place. That is where the organism is collected or observed. Ideally, the place is summarized by the global positioning system coordinates. But for, let's say, past collection in museum and also in history collection, you have a text description of a place, and there you have text description of a place, and you need to post your, let's say, your reference. But there, if you have to do this, you will have a problem of precision. So my advice to you is that when you go to a field, please keep your GPS with you. Take a GPS, okay, to record the coordinates. The third attribute is the time in terms of day, month, and year. Okay, those three attributes, let's say including the time, are important when you want to achieve completeness analysis, for instance. But for ecological niche modeling, okay, information on taxon, place, and also the last one and fourth attributes, basis for records are sufficient. So it depends on the analysis you have to do. If you want to achieve a completeness analysis, you will need information with taxon on the place, on the time, and also on the basis of records. Okay. If you want to achieve analysis or ecological niche modeling, you will need information on the taxon, on the place, and on the basis of records. Basis of record is the fourth attributes. Okay, it refers to the specific nature of the record and can refer to one of the six classes. That is, first is the living specimen. That is a specimen that is alive. Preserved specimen, a specimen that has been preserved. For instance, a plant on herbarium teeth, or cataloged loads of fish in a jar. You can also have fossil specimen, preserved specimen, that is a fossil, that is species that no more exists. You can also have material citation, a reference, let's say, to or a citation of a specimen in the literature, scientific publication, or PhD thesis, or master thesis. We can also have human observation, output of human observation. Let's say records that are directly collected on the field by, let's say, someone, okay, a researcher, or students, or naturalists, anyway, a scientist. And you have also machine observation, that are observation made by machine, recorded by machine, like a camera trap, for instance, information on camera trap, or film, or photos, etc. Now, where can we find the primary biodiversity data? What are their sources? First of all, let's say they are natural collection, natural historical collection inside herbaria globally and inside the museum. The information are found on specimen tags like what you have here. So this species was collected in Benin by Professor Akoenu Akpovi. He's, let's say, an elder professor. Now he's retired. And you can read the information. This plant, his family is Anacardiaceae family. The name is Surendia Warnekei. Okay, and here is the description of where it is collected. So this is a bush. A bush. It was collected in a bush in a locality, that is the village name, Hueto, Vidoto, for instance. And the date of the collection is a little hide here. Okay, it is hide it. Okay, you see. And let's say the observer or the collector is West Aquino. So those are the information you have to digitize in herbaria, history, natural history collection, and museum. Those are the information you need to digitize, and that exists. You have a natural history collection and museum also in Malawi. So to help publish more data from Malawi, you need to go there and find out, let's say, the information on the specimen tag and digitize them. Okay. We are also present, let's say, on specimen tags of fish, snakes, birds. You have snakes here. And let's say in the museum, from the museum, one museum of, I cannot read the name, but it is from the University of Copenhagen. And this is the name of the species, and the coordinates are also here. Other information, biodiversity, primary biodiversity data can also be derived from fossils and other paleontological material, like amber, that is, let's say, part of woods of plants that no more exist, for instance, with a specific yellowish color. We have also teeth or bones of animals, okay, or animal or plant samples, DNA, organs, specimen, for instance. You can also find primary biodiversity data, let's say in insectarium, that's a collection of insects. And here is the tag here, okay. The tag is here, okay, so that you can read and to digitize it. You have here specialists of taxonomy try to observe, let's say, organisms, specimen for their identification. They are quite serious about it, as you can see. We can also have data from literature, okay? Primary biodiversity data associated with multimedia, where information derived from thesis, master thesis, um, PhD thesis, or from scientific publication as well. So be careful. You have to pay attention clearly to the attributes. So the name of the organism, the plant name, most updated one, and we'll see on 10th, let's say during the course on data quality, how you can update the names, okay, of specimen. Okay, so in scientific publication, you need to pay attention to the attributes of primary biodiversity data. That is the name, updated one, but also the place where it is observed. Fortunately, some theses, okay, in their, let's say, appendices, they have the list of the species name or the animal names, and with the coordinates. If you have this, it is fine. But you can also read about, let's say, the test description of a place. As I told you, there is a problem of a precision when you have to post geolocate this. Okay, it is a problem. So the better is yes, to have directly coordinate with your GPS. Okay, other source of biodiversity of primary biodiversity data are field notes and records. For instance, from investigation or impact studies, okay, at a place where an enterprise or let's say industry has to visit, okay, species can be recorded to find out whereas it is more profitable to preserve, to conserve this part of a landscape or install, okay, let's say the industry. So the species records, okay, can serve as primary biodiversity data. We have also field notes, okay, field notes are recorded. Field notes are recorded directly, okay, on the field, okay, so that have right information, but you need to pay attention on the completeness of the information. Okay, we can also have participatory science data, for instance, citizen science. Okay, those can contribute a very valuable source of biodiversity, primary biodiversity data, but you need to pay attention to the quality of the data you have in hand. Okay, another source of primary biodiversity data are sample protocols. Yes, you go on the field, for instance, for an inventory, you have, let's say, you write the protocol, system, for instance, for a systematic sampling, as we can observe here, okay, systematic one. At each point, at each plot, you have a list of species that are assigned the geographic coordinates of the points. So they are quite valuable data, okay, to be cleaned and published. You need just to pay attention to the abundance data. Abundance data here. If you take, for instance, a record, in a given record, the same species can appear many times. But you have to take this into account only once. Okay, the number of the organism of the same species is here the abundance of the species. Okay, it can be useful, okay, quite useful in analysis, but you need to pay attention to just the presence of a species in a record and the absence of a species. Yes, also the absence of a species, but also the abundance of a species. Okay, absence of species that in a given record it is not there. Okay. Now, other source of primary biodiversity data is of is observational data, and it is the most, let's say, the largest quantity of data on GBU site actually. Okay, that is observation derived directly from the fields. Okay, on plants or animals during exploration, for instance, for citizen science that generated, let's say, a large amounts of information on birds, for instance. Okay, you can also associate images to, let's say, to the observation. Okay, over observational data can come from, let's say, animals, but if you are dangerous animals, and you need to pay attention, not to be too close in case we are hungry. Okay. So let's say you can take the pictures or set camera records and to have information on them. Okay. Now, with respect to those different attributes, what can we say? Okay, on the taxon, there are some issues you need to pay attention to. For instance, let us take the case of taxon observational data I just talked about. If identification is lacking or incomplete, no recourse is available. The record remains incomplete. What I ask my students to do is that when they go to the field, they need to take, let's say, samples of the specimen they observed. And when they return back, they go to the botanist or specialist of, animal taxonomy so that it can be identified. Okay. Otherwise, the record is lost if, let's say, a complete name is not, let's say, found, is not, yes, is not attributed when recording the material. We have virtual data, data based on specimen. So if identification is lacking, so for virtual data, if identification is lacking or incomplete, specimen can be re-examined or subjected to further study, for instance, from DNA barcoding. Inconsistent, you can also have, let's say, the case of inconsistent name. If identification is complete but not interpretable, for instance, for old name, synonyms, informal or incorrect name, that is local names, for instance, then nomenclatural research may be able to resolve identity in case of, let's say, uh, the existence of a specimen. With respect to the place, with respect to the place, you have GPS coordinates that are okay, fine, we talked about it, but you need information on the datum and coordinate precision. So the datum refers to the, let's say, uh, coordinate reference system. Okay, and what is widely used by GPS is the World Geodetic System 1984. Okay, in absence of coordinates recorded by GPS, you have text-derived coordinates that are useful, but Okay, you need full metadata to be interpretable. Okay, to interpret this. And what I can tell you is that it is a little difficult here because sometimes in a system of geolocate, for instance, you may lack some names. Okay, it comes that you record the specimen in a village, if you know a village does not exist in the system, so you refer for instance, to the municipality, the coordinator of a center of a municipality, and if the name of a municipality is not available, okay, you will need to refer to the coordinator of the department or even the country. So it is not precise at all. Okay. Anyway, coordinator from whatever source should be checked for consistency. Let us talk now about the time. The time in terms of day, month, and year are normal constituents. Month and year, or just year, are better than nothing, right? But the time needs to be checked for consistency internally. I will come later to that. And must also be checked for consistency externally if data are available and digital. We'll come to the example of this, okay? And I take example from the data of Malawi, animal data of Malawi published on GB site. I download this, and of course, we can try to find out what are the problem with respect to time, okay? Now, when we cross those attributes, for instance, you take the, let's say, the case of taxon plus place, you can have a description of a fauna and flora and of a country, of a given region. You can achieve typical ecological niche modeling, or we can also have gazetteers. Gazetteers are places with coordinates, okay? So if you have only places, you can have gazetteer, okay? Taxon plus places, let's say you can achieve it ecological niche modeling. And if you have only taxon available, you can then describe the fauna and flora of, let's say, your area of interest. Let us take the case of a place and the time. If you have the records in a place with time, then you can describe the sampling history. Okay. But if you have, let's say, the taxon records with time, then you can describe trend analysis. If you have information on the taxon, the place and the time, you can achieve completeness analysis. So it was the first part of the presentation. Okay, the first part of this presentation. And I will go further in a new file to talk to you about the second part.